Rice plants are prone to diseases. There are 4 known major diseases which affect rice plants in Guyana, but each of them can be controlled. Welcome to another edition of The Science of Rice, a weekly program brought to you by the Guyana Rice Development Board. I'm your hostess, Carol Gurusamy. On this week's program, we go in depth and learn more about diseases that affect rice plants and their control.
Basically, there are 4 major rice diseases affecting the rice industry in Guyana. These are blast, rice blast, Pyracularia griseae, brown spot, Bipolaris oryzae, and the sheath blight, which is Rhizoctonia solani, and sheath rot, Sarcoidum oryzae. All these diseases are of fungal origin. Basically, all the diseases that I have mentioned earlier, once we identify them in the farmer field, we will collect the samples and bring them in the lab for further processing and analysis in terms to confirm if based on the symptoms is the actual pathogen or the microorganism that we are thinking causing the symptoms in the field is the same in the lab. So what we do, we collect all the sample, process that in the lab, and then bring them inside for identification and doing the isolation.
The Pathology Department of the Burma Rice Research Station has qualified and experienced scientists to study and analyze the behavior pattern of each of these diseases. Once studies are completed, these scientists will be able to produce control methods and properly advise farmers about what to expect from their crop.
The objective of pathology department is simply, the first one is to, if I should go back a little to the breeding program. In addition to having high yielding varieties and varieties that can withstand lodging in the field and the harsh field condition, we have serious disease problem in Guyana over the past years, very long time back, like blast. Blast is the most dreadful disease of rice throughout the world. It could render plants very susceptible within a few seasons of plant being cultivated, and farmers could lose almost the entire crop, even at the stage of close to maturity.
Rice blast is one of the most destructive diseases and can easily be identified on rice plants.
To identify the blast symptoms in the field, basically you can look for what we call diamond-shaped lesions with an ashy gray center and dark brown margins. So this is what the center look like and the margin. The rice blast also would affect the panicle. So you'd have dark brown spots or lesions surrounding the neck of the panicle, and these are how it looks like on the leaf also.
Once the variety is not blast resistant, the rice plants can become vulnerable in contracting this disease.
We are developing varieties that are resistant to blast. And how we do that, we incorporate the resistant genes for blast into our local varieties. So all the varieties that we release, it is almost mandatory that they must be blast resistant. So the pathology department is very crucial to breeding also. As much as we are incorporating these genes into into the varieties, into the material, we have to identify those plants that are proving resistance in difficult situations. So the pathology department assists us in the breeding lines there. They will study the breeding lines in blast-prone areas we call hotspots, like in Von Better, in Crawford Creek, and Blackbush area, and in Kanji. And they are able to sort out which of those entries are proving good resistance, good level of resistance for us in Guyana here. And only then we look at the other traits. Because if it's not proving resistance, we will not go ahead with that entry. So it has a very key and pivotal role in the breeding itself. And apart from that, it looks after the general disease management of rice in Guyana, look for new options in terms of fungicide and so on for the use in rice.
While the scientists are working towards the development of these blast-resistant varieties, rice blast can still infest plants once conditions are favorable. But the disease takes time to break the resistance of the plant and spread.
Some varieties are resistant to blast and some are not resistant. Simply those that are resistant having the resistant mechanism to overcome that disease. So, and the one that is not resistant do not have that mechanism, or what you say, that gene to resist that condition when the blast comes. When blast comes when there's, it would coincide when there's high level of the blast spores in the air with good, maybe a little lowish night temperature, around 22 degrees, and a little morning dew. So these are conditions favoring the development of the disease. And probably a few brief shower in the day once in a while, you know. These are conditions with very high relative humidity, 85 and above. Those are conditions that are perfect for development of that disease. So if the plant is susceptible also, it will overcome it. As opposed to a plant that is resistant. Even though the conditions are good, but the plant is resistant, it will not be overcome by the disease. So that's how it operates.
There are three ways in which blast can affect rice plants. These are leaf blast, node blast, and panicle blast. Leaf blast symptoms affect the leaf as one can assume by the name. The leaf appears gray-green with dark green borders in the early lesions. In the older lesion, the gray spot is in the center of the necrotic border.
Blast is a very challenging and notorious disease to deal with. As of today, we are okay. And many countries around the world have noted the real devastating effects of blast. Varieties or plants that are resistant could later on render itself susceptible, because the blast disease itself, the fungus itself, would not stay in one form. It changes its composition, its genetic composition also, its whole composition, just to overcome that resistance itself. So while you are working against them, they are working against you also. So your challenge becomes even more now that you have to step up the level of resistance that you're putting in. Whereas 10, 15, 20 years, a moderate level of resistance will be good enough for blast. But now you'll have to have a very good level of resistance to be able to resist the disease, because it will not be left there alone. It's nature. So once you step up the resistance, they will step up also. So you have to be ahead of the game. Always ahead of the game. So we are ahead of the game up to this point, and I think we have mechanism in place to stay ahead. But it's not as easy as I'm saying it here. have shift gear in terms of breeding. And the whole perspective of breeding now, we have to take that, we have to look at the rice plant in context of the changing environment. And we have to develop the rice plant now in Guyana, the variety them, in the context of the environment in which it have to survive, not only survive and produce well. So there are adjustments have to be made at the minute level of the variety, and that's what we're doing, so that it will be able to stand up in the farmer field and give the farmer what it needs. We need a trial like this to run for at least 3 seasons, and it must be across the country, at least in 5 locations for 3 seasons. And after that, we'll select the best 1 or 2, and then go into the farmer field to 3 to 5 acre plot, and another 2 seasons of testing along with the farm at a larger scale, what we call that on-farm trial. And after then, we will able to pronounce along with the farmer if the candidate variety is ready for commercial release across the country. So from here, we're talking about 5 seasons from here.
These are simple cultural control methods for blast disease.
For the rice blast, all the variety that is being currently released in the rice industry is blast resistant. If you don't have resistant variety, then you should adopt the other cultural control methods, that is using disease-free seed, clean seed, or use your seed treatment for that's using the fungicide as seed treatment. Also, the disease generally survives on alternate hosts and plant debris. So it is advisable as a cultural method to get rid of all alternate hosts or plant debris that serve as residual or for the survival of the disease. Using the correct plant intensity, because if you have too high amount of plant or if you use a high seed rate, then you would have the ideal micro-environmental condition favorable for disease development.
While rice blast is one of the most important diseases in rice cultivation, brown spot is another common disease to look out for. Brown spot is a minor disease, but it is said to affect rice plants in the upland and wetland rice growing regions. However, if it is not effectively controlled, brown spot can cause great damage in a rice crop. Brown spot can affect a crop from seedling to mature rice plants.
The other disease is the brown spot, which is the bipolar ricey. It have similar looks like the rice blast. The only difference, the lesion actually are circular or oval in shape, and as the difference with the farmer basically confused with the blast and the brown spot, because the blast symptoms has pointed ends, or what we say diamond shape, but the brown spot is basically oval or elliptical in shape. So that's the difference between the lesion of the blast and the brown spot.
When the seedling is infested, the fungus on it appears small, circular, and brown. This causes discoloration in both primary and secondary leaves. The fungus can also infect the root as well, causing a black discoloration. Once the seedling is infested, it can stunt growth and even cause death. On the leaves of the older rice plants, the fungus has lesions that are more circular to oval and have a light brown to gray center. It is also bordered with a reddish-brown margin. When infection is severe, the lesions may merge, causing death in large areas of affected leaves. The fungus can also infect the glumes. This causes dark brown to black oval spots. Brown spot can also infect the grain, causing a black discoloration. These are all physical attributes of brown spot disease, but what causes the rice plant to become susceptible to infestation? Once there is a deficiency of required fertilizer for both plants and soils, also poor drainage, are all conditions that cause the plant to become vulnerable. Infective seeds, volunteer rice, rice debris, and weeds are all agents that can attract brown spots. Temperatures ranging from 25 to 30 degrees Celsius are favorable conditions for brown spot infestation. Also water stress and high humid conditions. If leaves are wet for 8 to 24 hours, infection can occur. Brown spot disease can be managed by following these simple steps.
In the case of brown spot, you need to ensure that you have correct nutrient application. That is, your NPK requirement in the soil should be at the optimum or at the correct level, correct all the deficiency, because in general we see an increase in incidence of brown spot when there is a deficiency of either the P and the K in the soil.
We applying our nitrogen fertilizer, which would be our first application of fertilizer, which done around 18-21 days. And we applied the nitrogen fertilizer on dry or moist soil, as you see it in the field already, how the field is. So we're using a mechanical spreader, and the rate that we're using here is 110 pounds per acre urea.
And the reason why it's being applied on the dry soil or on the reduced water level is because you want to prevent nitrogen loss. That's the primary reason why we are applying the urea on the dry soil. In the past, urea used to be applied under flooded conditions. It has been established that when that happens, or when that takes place, over 50% of the nitrogen is being lost due to ammonium volatilization. Basically what happens here is the urea breaks down, and the nitrogen, the urea breaks down in the water, and the nitrogen escape back into the atmosphere, and studies have shown over 50% of the nitrogen can be lost.
Other than that, you keep your surrounding clean for alternate hosts and so on, and the other alternative, if there's a disease incident and you have no other option, is to go for the chemical control. And the two chemical we are currently recommending is the Fuji-1 at a rate of 200 to 300 ml per acre, and two motor blow-off spray solution per acre. So you divide that amount of chemical in the two spray solution, and the other one is the carbendazole at the same or similar rate.
Other control methods used against brown spot are: 1. Avoid water stress conditions. 2. Sow resistant varieties. 3. Keep field and access dams clean and weed and debris free. 4. Brown spot is a seedling disease, and it is advised that farmers should soak their seeds in hot water seed treatment 10 to 12 minutes before sowing. Sheath blight is another disgusting disease to deal with. This disease affects rice cultivation under the irrigated or wetland system of production. Sheath blight is a soil-borne disease, and pathogen can spread rapidly through irrigation water. It can also spread in the soil during land preparation. The symptoms are very vivid to the naked eyes. It appears when plant is in the late tillering stage.
Sheath blight basically appears about 3 cm above the water line. as grayish, brownish water soak lesion at on the tiller at the water line.
Sheath rot is another disease that can cause losses. However, in Guyana, the losses are small scale, especially with the new technology used in rice cultivation and the high quality varieties produced.
The sheath rot symptoms would show up on the flag leaf of the plant as dark brown discoloration on the flag leaf.
Another common disease is fall smut.
Fall smut is appear as a dark brown velvet color on the grains when the rice is in the reproductive stage, and as it get older, it get dark brown, dark black to brown, and in the earlier stage it's kind of yellow.
Sheath rot and sheath blight have the same cultural control methods as brown spot. Research is done on seeds to determine whether they are vulnerable to any of the 4 major diseases before they are sown in the fields.
Currently in progress is the seed health testing. Here we have collected seeds from the different rice growing regions in Guyana, and they are being placed under a test, that's the moist test, to ascertain what fungus will grow on these seeds, if any. Here we have fungus being grown, and basically what you see here is alternaria, which can lead to the rotting of seeds when it is sown. So our job here is to test these seeds and then report to the farmers and let them know the seeds that they were intended to use as seed paddy, they have the fallen fungus on them, so that they can take precautionary measures as to treat the seeds with fungicide before sowing.
Tests are also done on seedlings.
Okay, these seedlings were taken from the seedling vigor study, where we use the seeds, disease seeds from the 4 important diseases of rice, such as blast, sheath blight, sheath rot, and brown spots. Now, what we are looking at is to evaluate the impact of the disease on the root and shoot length. Also, we're looking at the impact on germination and establishment in the field. So the seeds are taken and they're brought in, and then we would measure them, right? The root length and the shoot length. And we are going to put them on the evaluation and analyze the data and make recommendations to farmers that if they use seeds that are taken from a disease plot, these are things that can be expected to impact the rice crop, the current rice crop.
Testing of insecticides is also carried out when tackling diseases in rice.
In the procedure being conducted here today, it's being done on compatibility study, where we are putting several plants under the trial of testing them against fungicide, weedicide, and insecticide. Now, these cocktails are very common in the field. Farmers will mix them as a result of cutting costs. And today, we are actually running the test to show the farmers the impact of mixing such cocktails and applying it to the plants. Regional. And the rate of application here is, well, since we are mixing it for 500 mLs, we are applying 0.3 ml for the 500 ml liter water.
Chemical application is the last method of control. Once farmers can practice the cultural controls as mentioned earlier, they can have these diseases under control without the need of applying chemicals. Ensure you monitor your crop closely and implement these methods when necessary. Thanks for joining us on the Science of Rice. Do remember to join us next week as we continue to journey through the rice industry in Guyana. I'm your hostess, Carol Gurusamy. Do have a pleasant rest of the night.